So first of all, four very nice presentations. And my question actually first goes to Sehan. So as an immunologist looking at what you did, I see that, the collaborator that you had there gave you 14 different subsets of cells to look at. But when I look at a CD8 cell, and I explore those phenotypically with all 400 known CD markers that have been clustered from the HLDA, I know that there's at least 195 different proteins expressed. So if you only go to the depth of a CD8 cell as you did, the complexity of the phenotype is so different in there that you would have to at least probably go through maybe 20 to maybe 50 million cells before you ever be able to, get the data. So how do you conclude the data out of there if it's not mapped to a much more defined phenotype?
Well, I think this is just a start of this. Like I see, and I agree with you that that what we have identified as cluster and named as CD8 or CD4s needs to be we have to go more deep into it, and for that you need more cells, as you said, to be able to differentiate all these depending on. So I think this is just proof of, as I said in the beginning of my talk, this is proof of the principle, but it is possible. And we have to remember that this is a very new technique, and it's continuously developing. And so there will be a day, I believe, that we will be able to identify millions of cells only in one individual. But we are not there yet, but there will be a time coming up. But these studies are good in a sense to actually develop the statistical methods and how we're gonna handle this data in the long term. And of course, the other thing that what we need to do is we have to do more functional studies and look at whether that's the SNPs has a effect on the cell types that we are actually thinking that it has an effect, and that's something that we are also considering during with CRISPR technologies, to be able to make a switch and then see if that is actually have an effect on the cell gene expression for that specific cell type.
Okay.
So there are ways to to explore more.
It just seems to me that with the site-seq technology that came from Mignotti's lab at the New York Genome, that combining that today, you can put 200 to 400 antibodies that are that have been conjugated with oligos.
Yes.
And by doing that, and you combine it with your WTA, then you'll at least get there in a way that's much more exact, because I don't think you could probably just get there with the gene.
I agree, and that's another aspect of this study. So we although I haven't told you the whole story about this 1K project, so we collected blood samples, a lot of blood samples from these 1,000 people, and we have so much extra samples for each individual, so we can actually do some additional analyses. And site-seq is one of the other experiments that we are actually going to do in the next year or so. So then that will be another level of information. But I think one thing that I always I think what we are seeing at the proteomics level could be totally different to what we are seeing in transcriptomic levels. So I don't think we clearly understand the switch between what's happening at the transcriptomic profile versus the protein profile. So and there will be more studies required for us to understand that better.
All right, a question for Al for us. So, firstly, commend you on doing this actually and having the funding to, glad to see someone's doing this massively parallel comparison. So I was wondering, confused about something. So cryo versus fresh makes sense in terms of, say, stress response genes and, of course, recovery of cells, and therefore you lose some in cryo. In terms of warm versus cold, I was wondering, so in terms of dissociation, so why is it worse in warm compared to cold? Because you're keeping it still fresh at 37 should be more
Yeah,
physiological.
So at 37 degrees, you're transcriptionally active. So all the stress, and typically they're in the order of an hour, or it depends on the protocol. There's so many different flavors of dissociation protocol out there, but basically these are transcriptionally active, responsive cells. Now, if they've been in a tissue and then you're suddenly pulling them apart, they're going to freak out, and that's what you see. And this is what Stephen Potter published 2 years ago, a year and a half ago. So, yeah. But then you've got which is best is a different question, because there may be cell types that you get a better representation that truly matches, because you need the warm dissociation to release them. The cold dissociation maybe hasn't released them fully. So we have a hunch that, I can't remember which one, I think it might have been the podocytes. There's one cell type where we see lower actually Liber Henley, there's lower in the cold dissociation. Now, it could be that is closer to the ground truth, or it could be that those cells are harder to dissociate in the cold condition. It may be we need to extend the cold dissociation. There are protocols where people take some off and then whack some more protease on there and let them keep on going. Mm-hmm. But I think we really need a ground truth to get at it properly.
Right. Just a quick follow-up. So you do see this difference between immune and epithelial cell recovery between the single cell and the single nuclear RNA-seq, so at least in kidney. So is it similar in other tissue types as well? Have you tested?
So the stuff that came out of the Broad Lab with the single nuclei, single RNA toolbox, admittedly it's on tumours where they've split them into 2 bits, done one with nuclei, one with single cell, and they do see depletion of the leukocytes. So it's not macrophages. So other immune cells are happy, but Chris Wells is smiling there. But the leukocytes are massively depleted. I don't know, has anyone in the audience got good recovery of leukocytes from single nuclei methods? Okay, no one's putting their hands up. Okay, cool. Cheers.
Have you considered spatial approach to benchmark?
We're going to play with, yes, basically yes. The problem is then you're talking a Z stack, and that adds massive amounts of money to do the experiment properly. So yes and no.
You know, I can answer your question about the nuclei.
Yeah.
So what it really depends upon is the way that you isolate the cell. And even in cell sorting, there are 2 specific types of sorting technologies that are used in jet and air, which has an acceleration of the cell from one position to another for its sorting, and another that's based on a fixed nozzle where the cell tumbles through. In each case, depending upon the type of cell that you're using, there's an impact to viability. And at the nuclei side, then it can have a huge impact on that.
So just to comment, so one of the I didn't mention, but one of the nuclei protocols we used doesn't use fans at all. So it uses a sucrose gradient. So we're not doing a sorting step for those, and we see the same bias.
The impact of sucrose is huge on a cell.
Maybe to Jay or Jonathan, what are the best practices for integrative analysis of single-cell ataxic and single-cell anergic at present?
Sorry, I didn't catch the question.
Okay.
You were asking about the best practice?
Best practices to integrative analysis of single-cell RNA sequencing and single-cell ataxic.
Together or separately? Together.
Yeah,
what is the best practice? So we are aware that there's 4 technologies out there, including ours, ISTASIC. There's also a technology developed by BGI. There's also a technology developed by Kun Chang's group, as well as the technology developed by Jay Shedden's group. I think they are very different in terms of the number of cells they want to profile and how deep they want to sequence. That's one. Second difference is some of the group, they were focusing on nuclear mRNA. As Alistair had already alluded to, when you look at total RNA or nuclear RNA, single cell seq, there's a difference. And I'm also aware that some other groups, they separated the nucleus and the cytoplasm at the first steps. So basically, if they do that, they will focusing on cytoplasmic mRNA portion. So for our technology, we focus on total mRNA. So I don't think there's a best technology out there yet, but I think it's up for the field to really test them out independently. Yeah.
Okay, we will allow for one last question.
Okay, a quick question to Jay. How about using RNA fish to detect enhancer RNA? Do you think it can be a good complementary information for the 10x5' sequencing?
Yeah, so it is possible to detect enhanced RNAs using single molecule fish. So the textbook knowledge of the length of enhanced RNA is 300 nucleotides, but that's not always the case. So there's actually no good comprehensive annotation of this enhanced RNAs. I think someone should do full-length nanopore or PacBio on these enhanced RNAs to get really good annotations of these enhanced RNAs. Actually, if you do, then you can actually use 3' strategy to detect enhanced RNAs too. But nevertheless, I think you can, as long as you can design 15, 20 fish probes against these enhanced RNAs, you are able to detect them. What I usually recommend is to also co-label with nearby protein coding gene at the intronic region. So what we tend to see is co-expression of nearby promoter gene, protein coding gene, and enhanced RNA should overlap somewhat nicely. So that's kind of how we prove the detection.
Do you think it's feasible to do it at the genome-wide scale, like to detect 10,000 of enhancer RNA?
Yeah, you could consider like sick fish or sick fish plus from Long Kai's lab to do enhanced RNA. So we are interested in doing this for non-coding RNAs in general, but we can maybe perhaps push that boundary to enhanced RNAs. Yeah, thanks.
How many cells do you need to average over to get decent annotation? And does it saturate, like after 2,000 cells, does it saturate or something like that?
Yeah, so the number of, it saturates similar to the promoters. We do have to have pseudo-cluster, like cluster of cells, in order to identify cell type specific enhancers, for instance. But the total number of enhancers are, still relatively substantial that we're able to make a lot of these correlative analysis. Of course, there's a lot of room for improvement to increase the capture rate of enhanced RNAs, but I think that will be kind of synergistic to detecting promoters as well. So that front needs to be further pushed to increase the sensitivity for sure.